Good evening, everyone. It's my great pleasure to welcome you all on behalf of IADWEL Kerala Research Presentation Cell. Today's session is on Cochrane Review. Cochrane is a British international charitable organization. The Cochrane Library and its collection of Cochrane reviews is a key source in evidence-based medicine. The review provides valuable guidance for clinicians and patients in making informed decisions about treatment options. SJSTN is a condition with high mortality and its management can be challenging for clinicians. We'll be discussing today the latest Cochrane review of the available evidence on the effectiveness of different interventions for SJSTN. Ajit sir, Professor and Head Department of Dermatology, Government Medical College, Cochrane, will be chairing the session. As you all know, sir is a great teacher, clinician, and a researcher par excellence. Sir is also the convener of IADWEL Special Interest Group in Research. Welcome you, sir. The Cochrane review will be presented by Dr. Farzana, a postgraduate student in dermatology from Government Medical College, Cochrane. Welcome you, Dr. Farzana. I would also like to extend a warm welcome to all the seniors and other members and postgraduates who are with us today. And I'm requesting the senior members to share your insights on this review after the session. I hope that this discussion will be informative and engaging and will help in improving the care and outcome of patients with TN. Thank you. Ajit sir, please.
Good evening. Thank you for that introduction. Actually, I'll start with a conflict of interest statement first, or disclaimer rather, that I'm not a great expert in this field, nor I have done any meta-analysis or Cochrane review. It's not part of that. But since my colleagues and friends insisted that I should do this, I thought, okay, I will do it. Just introduce this, what I know, and we will discuss it with fellow IADWELites. This program is planned like with the 3 sessions. Initially, I'll just give an overview of what is Cochrane review. Then Dr. Farzana will present one of the latest Cochrane reviews. Then we will have a general discussion on whatever Dr. Farzana presented. That will be our plan. So please be active in the discussion. I think this is an important area because every day the way we approach evidence is changing, and especially with the advent of AI. Probably in few months' time, the way we look at literature is going to change because see, even now, if you give a clinical problem, AI will give an answer with all the differential diagnosis and just like a discussion of postgraduate discussion. We'll get the summary of the whole thing. That's the scenario now. So the whole way we approach clinical problems is going to change in next few years. So it's very important to understand how the evidence is being, is being compiled by us and how it can be used. Unless we know this, we may not be able to compete with the machine in next few years. So let me share the slide. And one more thing, SIG DCR, that is Clinical Dermatology Research, is conducting a one-day workshop on meta-analysis by experts, actually Cochrane review by experts in Bangalore on 28th of May. And registration is free, but it is not online, it's offline. So welcome for that. It will be at Ambedkar Medical College, Bangalore. Is my presentation visible?
Yes, sir, it's visible.
Okay. So why Cochrane review and what is Cochrane review? That's what I'm going to discuss in next 10 minutes as an introduction to the presentation by Dr. Farsana. Is for those who are not very familiar with Cochrane review. Maybe it is very superfluous for seniors and experts. Please pardon. Why Cochrane review? As Dr. Sabina mentioned, it is to find out the best available evidence regarding a clinical question. Cochrane review addresses clinical questions for which we are looking for answers. And again, as mentioned here, it's a non-profit organization that aims to help people to make well-informed decisions about healthcare. By preparing, they prepare, maintain, and promote systematic reviews. And they keep doing it. So a systematic review done now will be reviewed after a few years and updated. So that it gives the updated information about the current evidence. And this helps to update us on the healthcare, the best healthcare interventions. So that's the purpose of Cochrane review. Cochrane library is freely accessible for Indians, thanks to Government of India, who pays a huge amount for that. So it should make sure that we are making use of this. It's accessible, not because Cochrane Collaboration is giving it free, but because the Government of India is paying for that. As far as evidence is concerned, we know the evidence is changing every day. New information's coming. We are in the era of a Big Bang, knowledge Big Bang, information Big Bang. So it keeps changing. And we read different studies, we'll get different results, we'll see different results and different conclusions and different claims. So how to make a sense out of this? How will we find out which is right, which is wrong, how much is applicable to a situation? This is what Cochrane reviews will help us in. This is something very familiar to many of us, the evidence pyramid. There was a time when the professors and HODs used to utter gospel truths, but not now. The expert opinion is the lowest level of evidence. Just because somebody is considered to be an expert, the information need not be correct. It's the lowest level. Then case series. Okay, experts say, okay, I have seen one case. In my vast experience of 3 cases, I saw this. That is expert opinion. Case series and case reports. A little more better because it is documented properly. Maybe more than one case many a times. So there's a trend at least, but it gives at least, it gives rise to a question, even though it doesn't give an answer. Then larger observational studies with comparison groups. There's one group with the treatment, the other group without treatment, or one group with one intervention, the other one without other intervention. That's next level. Then control trials. So you are intervening and giving some medicines, but the subjects are not randomized, not randomly distributed between the groups. That is next level. Then RCTs, randomized control trials. Many of us stop at this level. But what happens is if you do 10 studies, 10 RCTs may give 11 different types of results. So again, difficult to make a sense out of that. So that's what systematic reviews do. Systematically, you collect all the reviews and compile it and give in a systematic way, review these articles and compile and write it up and try to make some conclusion out of that. When we go to meta-analysis, we pool all this data appropriately, systematically, and do a statistical analysis of the data and practically write another paper, putting all this information available from different systematic reviews and other information if it's available. Cochrane review is actually a meta-analysis. Now what's happening is there are many people coming out with meta-analysis, even many of them with bias. Even medical companies and special interest groups produce their own meta-analysis. So Cochrane Review is an independent non-profit organization which try to very systematically, even they prescribe the systematic way and softwares and methodology to do this meta-analysis and keep on providing meta-analysis available online, and many of them are published also. That's what Cochrane Review does. That's why it is at the topmost level of the evidence pyramid. So this is what Cochrane Review does. For example, for SAST, which we are going to discuss, it's a rare disease. So the evidence will be rare, also will be rare. The evidence of treatment efficiency is limited because, again, we may see 10 cases maximum in a year, even in a big center. Most of the places maybe 1 or 2 cases or less than that. And in a study, in a practice survey, they surveyed the practitioners. What they found out is only 50% of the physicians reported that they are following treatment guidelines or an institutional standards even. And only minority of these physicians use professionally published guidelines. So this is the situation, and the evidence, whatever evidence we get from this also will be like this only. Because there are very few cases goes into any kind of trials or studies. So it's very important to get to know what is the current evidence from whatever is available. So why meta-analysis? I already discussed this, to establish statistical significance with the studies that have conflicting results. So there are different studies with the conflicting results to put everything together and get a statistical significance for the outcome. To develop a more correct estimate of the effect magnitude. So different studies are there, different RCTs are there, different observation studies are there. We're giving different effect magnitude, different numbers. So putting everything together and give a more correct estimate. To provide a more complex analysis on the harms of the intervention, not only it gives it analyzes and finds out not only the effect of intervention, but the harms also, and about other benefits also. We will see this in the example which we are going to discuss. So how to do that? So we have different RCTs, observational studies, many studies are there. So using this, we synthesize data and summarize and get an estimate. So there are 4 important steps for this. One is, first, you'll have to identify all the available studies. That's the first step. So if you are planning to do a Cochrane review and synthesis evidence, first thing is to identify studies. You have to get all the studies on there. And then define eligibility criteria. You may get 50,000 studies if you do a literature survey on some topic. But which all studies are eligible to be considered for analysis? So you should define that. You should make a definition for the eligibility. So you collected all the data from all these eligible studies, then abstract the data from that. Whatever data that's there published or available, get all the data, then redo the statistical analysis, putting all this data together. That's what we do in a meta-analysis. So we'll go to a little more details on that. I'm not a statistician, but still we'll try to just have a general idea how to do that. Okay, first thing is to identify studies. Okay, maybe there are some studies to start with. There are some studies in your computer itself which you read recently. Then you got interested, so you read some 5 or 10 articles on the subjects. Collect them. Then there are n number of electronic databases like PubMed, Scopus, Embase, etc., etc. So collect all the articles or at least abstracts from all these databases. Then go to the references of these important articles and look for any articles which we missed, but is referred in these articles. Then you can contact experts who are doing trials or studies. Probably they have some data available with them, which is not published or being published or like that, or rejected like that. So collect those data. And if there are any new articles which is being published in the very recent days, collect that also. So this will give you a huge number, many times a huge number of articles. Then you'll have to evaluate the quality of these studies. We won't analyze every study or compile every study. We'll just evaluate the quality of the studies and find out whether these studies are good or not. And fix the eligibility criteria regarding the studies which we should include in the study. So most important thing is to define the search strategy. What are we going to? So interventions for SJSTEM. Will you include all different studies, every studies, or will you include only English studies, or you include French also? Are you going to include from India and neighboring countries only? Or are we going to study only Western studies? Or are we going to select only from 3 important electronic databases? We'll have to fix that. We'll have a clear search strategy. These databases also have a lot of limitations. Many of them will be incomplete or may have imperfect. Language can be a problem. Problems with or sort of gray literature can be there. Many countries have their own data which is not published. Many organizations may have their own data which is not published. Maybe there are program data which is not published but is available in program documents. So all this kind of data have its own problems. Then there's something called publication bias. Negative studies are unlikely to be published. You have a big study, but it doesn't show any positive result. We may not send it, even if sent, many journals may not accept it. So just like that, there are various factors which can lead to bias. If there are 10 studies recently published, it's very unlikely that an 11th study will get published, even if the results are a little different. So that kind of bias can occur even in publication. And in countries like underdeveloped countries, things may not get published, even if they do a good study. So how to deal with this kind of bias also is an issue. So even if you collect all the data, there can be a bias. So dealing with the bias publication is important, and research strategy is very, very important to deal with that. Then, so we have all the studies with us. then define the study quality criteria early enough. So you should have a scoring system to decide the quality of the study. So you should decide, okay, we'll take these scoring, use this scoring system to assess these studies, and only those studies fit into this will be selected for the analysis. Then abstract data. So you collected all the studies which are relevant and which have a quality, reasonable quality, fits into the strategy. Then extract the data from that. For that, first identify the relevant articles, sort out the eligible articles, set up, make a form. We'll show, Farzana will show how to do that in on a small example. Then set up a form for abstraction of the data. Then enter all the data from these eligible studies, do a statistical analysis. Then when you do that, you should decide what all things you are going to analyze. Are you going to analyze the mortality only? Are you going to analyze a specific morbidity? Are you going to analyze a long-term morbidity? Or going to analyze the duration of admission, the hospital, or time taken to get better or get discharged? So all these things you should decide first. What are we going to study? So what kind of evidence are we going to make? For example, are we going to see a particular intervention? Like, okay, IVIG is going to make any difference or statistically significant difference in the mortality or duration for discharge or long-term side effects like pulmonary complication for due to Stevens-Johnson syndrome. So it should fix this before starting the trial, starting the analysis. Okay, these are the things which we are going to look at. And what measure are you going to analyze? Is it an odds ratio, relative risk, proportions, or whatever it is? Define that. And what kind of effect model you are going to use? Is it only limited into a limited population or in a random generalized population or whatever it is? Then do the analysis and also make sure that there is homogeneity or there is homogeneity between the studies. All the studies look alike or the similar studies or not. Because the studies are different types, that may not have a homogeneity. I'm not going to the details, just to go through to make it simple. So once you have collected all the data and decided what are we going to do, then you'll have to decide what are the measures which are going to look at. I already discussed this, whether it's odds ratio, rate ratio, whatever. You have to decide what you are going to look at. Then also you should look for the homogeneity we already mentioned and random effect model, etc. Then what we'll do is to see what's the publication bias. So each study will have a particular number of measure of effect and a particular sample size. So put this into a graph, and if the graph looks like this, like a funnel, that means there's not much bias in the publication. But if it looks like this, that means there is a publication bias. There is some issue with at least some of the articles published. It's a bias. So you may have to use this information with a pinch of salt. And the last thing is something which when we go to meta-analysis or Cochrane, you often see there's something like a forest plot. Forest plot compiles the whole every study which is studied. So, for example, this meta-analysis had maybe 8 or 9 studies. This shows the numbers, the positive result, and the numbers included for treatment and control. And here it shows the relative risk and the confidence interval. And it is diagrammatically represented here or schematically in a graph. So the first study, the risk ratio is 0.71. Okay, so it's 1, 0.5, 0.71, and the relative risk is confidence interval is 0.39 to 1.31. It is crossing 1. So the significance statistically it's not very significant. But see here, it is only on this side. That means it has some positive effect in this. I don't know what the study is about. It shows that the study shows a positive effect. If it's only here, it's negative. And this diamond shows the overall statistic of the study. Okay, there's a total effect size. It shows the average confidence interval and size, etc. So this gives the details. So this is the summary of this Cochrane review. So, so this is in summary how Cochrane review gives some idea. And ultimately, so we will say that putting everything together, there's a strong evidence to say that, okay, this intervention is useful, or this intervention is useful. Or many times we will say that actually there is no evidence to say either. So knowing this, we'll be able to make evidence-based decision in a clinical scenario. So we will discuss that after Hosanna discussing this systematic review on SJSTEN, how to use that information from that study in a clinical scenario. So let us discuss an example of a systematic review, Cochrane systematic review here. Sorry, I think I took more than 10 minutes for that. I think can systematic Farzana start presenting, Dr. Sabina, Dr. Mary?
Yes, sir. Yes, sir. She can start presenting.
Can you share?
Farzana, can you start slide share?
Okay, madam. Is my screen visible, madam?
No.
No, Farzana.
Share my screen.
Any issue? Any problem?
Actually, I can't share.
Farzana, can you share slides?
There's a problem with the net. You can you close the you can close the video and try to share.
There is some issue.
Then, should I share, Farzana? Should I?
You can,
Your net is very low, I think.
yeah, you can close the
Yes,
video.
madam. Can you please share?
Please close the video, Farzana.
I'll log in once again. I'll log in once again, sir.
Okay.
Okay. Actually.
Farzana, your screen is visible. Screen is visible. Please start slide share.
Is it visible, madam?
The screen is visible, but slides are not visible.
Slide is not.
Please start slide share.
I'm already shared, madam.
Oh, but it's not visible here.
Yeah. Now it's coming.
Yes. Now it's.
Is it moving now?
Yes.
Yes.
So you can change to slideshow.
Please close that and start slideshow.
Now.
Slides are visible, but slideshow is not there. Some window is open there. Please close that window and start slideshow.
But it is.
That activation wizard,
It is.
close that.
Please close that.
But it is in slide.
Yes.
Okay.
It's
Okay.
now.
Yeah. Okay.
Now you can start slideshow.
Okay. You can switch off the video.
Yes. Yes, now it's okay, Farzana. Now it's in slideshow, you can start. Ms. Farzana, please unmute and talk. You are not audible.
Am I audible now?
Yes, now you are audible. Please start from beginning. You are not audible initially. Yes, Farzana, you may start. You are not audible now. Farzana, you are not audible.
Hello?
Farzana, can you hear me? Farzana, can you hear me, Farzana? Farzana? Yes, now you are audible.
ഇപ്പൊ കേൾക്കാവോ സർ?
Yes, കേൾക്കാം.
Good evening all. Here I am presenting a recent systematic review published in Cochrane database on systemic interventions for treatment of Stevens-Johnson syndrome, toxic epidermal necrolysis and STS-TN overlap syndrome by Jacobson et al. STS-TN and STS-TN overlap syndrome are rare, severe cutaneous adverse reactions usually triggered by medications. In addition to tertiary level supportive care, various systemic therapies have been used, including glucocorticoids, intravenous immunoglobulins, cyclosporin, N-acetylcysteine, thalidomide, infliximab, etanercept, and plasmapheresis. There are a lot of controversies regarding the treatment for this potentially lethal disease. So there is an unmet need to understand the efficacy of these interventions. So here we are going to discuss currently available evidence regarding the management of these conditions. The objective of this review was to assess the effects of systemic therapies, including medicines delivered orally, intramuscularly, or intravenously for the treatment of STS-TN and STS-TN overlap syndrome. They have searched databases up to March 2021, which includes the Cochrane Skin Specialized Register, Central, Medline, Embase, 5 clinical trial registers, and reference list of all included studies and of key review articles, drug manufacturer websites, and search for errata and retractions of included studies. The 5 clinical trial registers included were ISRCTCN Register, clinicaltrials.government, Australian New Zealand Clinical Trial Registry, WHO International Clinical Trial Registry Platform, and EU Clinical Trial Register. Types of studies included were randomized controlled trials and prospective comparative studies, irrespective of language or publication status, which means published, unpublished, in press, or in progress studies were included in this review. Participants of any age with a clinical diagnosis of STS-TN or STS-TN overlap syndrome were included in the study. STS-TN were classified as per published criteria by Bastuji Garin et al., but included all studies reporting a clinical diagnosis of STS-TN. Types of interventions included were all systemic therapies studied to date, including corticosteroids, IVIG, cyclosporin, N-acetylcysteine, thalidomide, infliximab, plasmapheresis, and etanercept, and comparisons between each of these therapies, and also comparisons of some therapies versus placebo. In the protocol, the authors wanted to analyze primary outcomes like disease-specific mortality, adverse effects leading to discontinuation of therapy, and secondary outcomes were time to complete reepithelialization, ICU length of stay, total hospital length of stay, illness sequelae, and other adverse effects attributed to systemic therapy. In effect, they wanted to analyze about the morbidity, mortality, and adverse drug reactions. For that, they have created a Prisma flow diagram, which will show in next slide. It shows how they selected the studies for this review. And if there are multiple reports from a same study, they collated these reports and considered it as a same study for making this diagram. This is the flow chart in which initially they identified 6,116 records through database searching, and there were 6,077 records after removing the duplicates. And 5,000 while screening the titles and abstracts, 5,974 records were excluded, leaving 103 full-text records. And among these, 45 full-text records excluded, 44 due to which had wrong study design, and 1 due to ethical and data integrity concerns. And 43 additional studies were also excluded due to when they developed these exclusion criteria. And other studies removed. 3 were ongoing studies. 1 study was awaiting classification. And finally, they assessed 9 studies in qualitative synthesis and 2 studies in quantitative synthesis. So the studies included were 3 randomized controlled trials, 6 prospective controlled observational study, and 2 studies in the quantitative meta-analysis. There were 308 participants with 131 males and 155 females from 7 countries. The study settings were 2 studies were conducted in burns units, 2 were in dermatology wards, 1 in ICU and 1 in a pediatric ward, 3 were in unspecified inpatient units. Most of the studies did not report study duration or time to follow up. 7 studies reported a mean age which ranged from 29 to 56 years. 2 studies reported a mean Cochrane of 3 and 1.9. And 2 studies included pediatric participants with a total of 23 children. Even though the authors wanted to analyze interventions like etanercept versus cyclosporin, etanercept versus IVIG, IVIG versus supportive care, IVIG versus cyclosporin, and cyclosporin versus corticosteroid, they had data to analyze only these interventions, which were corticosteroid versus no corticosteroids, IVIG versus no IVIG, etanercept versus corticosteroids, cyclosporin versus IVIG, N-acetyl cysteine and infliximab versus infliximab alone, thalidomide versus placebo, and plasmapheresis versus other treatments. After selecting these studies, they evaluated for the risk of bias in these studies. They assessed risk of bias for each outcome by using a software called ROP2. By using this ROP2, they tabulated bias like bias arising from randomization process, or bias due to deviation from intended intervention, bias due to missing outcome data, bias in measurement of the outcome, or bias in selection of reported results. And they finally arrived at a comprehensive judgment about this bias of these results in each studies. For example, if a study with serious risk of bias, they mention it as a red color in the table. And if with a moderate risk of bias, which represented as a yellow in table. And if a study with low risk of bias, which represented as a green in table. So in our study, there were 3 RCT. Among these, 1 RCT, results from 1 RCT assessed as low risk of bias in all domains, and results from another RCT as high risk of bias, and from 1 as some concerns. And results from all 6 prospective observational comparative studies to be assessed as high risk of bias. Next, after doing this risk of bias, they evaluated for the certainty of evidence by using grade consideration. Grade means grading of recommendations, assessment, development, and evaluation. To avoid the risk of bias in certainty of evidence, each article was independently assessed by this different authors. And the 5 areas considered was, first one, risk of bias, which we already discussed. Second is imprecision. Imprecision is mainly focusing on the 95% confidence interval. So if the certainty will be low if a clinical decision is either at lower or upper end of this confidence interval. Next is inconsistency. Inconsistency in which certainty will be highest when there are several studies that show consistent effects. Next is indirectness. Evidence is more certain when studies directly compare the interventions of interest in the population of interest and report the outcomes. And publication bias will be more for observational data and when these studies are funded by industries. These are the 5 areas which considered in the grade consideration. And this certainty of evidence assessed as very low, moderate, or high. And from evidence from RCDs assessed as high quality, and evidence from observational studies also starts at high quality. So overall, in this systematic review, overall there is a high risk of bias with high certainty of evidence. And finally, the authors could analyze only these interventions like corticosteroid versus no corticosteroids, IVIG versus no IVIG, etanercept versus corticosteroids, and cyclosporin versus IVIG. That too for pre-specified outcomes like disease-specific mortality, adverse events leading to discontinuation of therapy as primary outcomes, and secondary outcomes were time to complete reepithelialization, ICU length of stay, and total hospital length of stay. So in final conclusion, when they comparing this corticosteroid versus no corticosteroid on the effect on disease-specific mortality, which was uncertain, and time to complete reepithelialization, length of hospital stay, adverse effects leading to discontinuation of therapy were not reported. And about the IVIG versus no IVIG, effect on disease-specific mortality, time to complete reepithelialization, or length of hospital stay was uncertain. And adverse effects leading to discontinuation of therapy were not reported. And when comparing the etanercept versus corticosteroids, etanercept may reduce disease-specific mortality. And sepsis and respiratory failure were reported in 5 of 48 participants with etanercept and 9 of 43 participants with corticosteroid. But it was not clear if they led to discontinuation of therapy. Time to complete reepithelialization and length of hospital stay were not reported. And about the comparison between cyclosporin versus IVIG, effect on disease-specific mortality was uncertain, and time to complete reepithelialization, length of hospital stay, and adverse effects leading to discontinuation of therapy were not reported. And about the comparisons between N-acetylcysteine and infliximab versus infliximab alone, and thalidomide versus placebo, plasmapheresis versus other treatments. These were small studies, so no conclusion could be drawn from these studies. So overall in conclusion, when compared to corticosteroids, etanercept may result in mortality reduction. For the following comparison, the certainty of evidence for disease-specific mortality is very low. That is for corticosteroids versus no corticosteroids, IVIG versus no IVIG, and cyclosporin versus IVIG. So there is a need for more multicentric studies focused on most important clinical comparisons to provide reliable answers about the best treatment for SGSTA. Thank you.
Sabina, madam.
Can you close this slide share?
Yes,
Okay,
sir.
okay.
Yes, sir.
Okay.
Ajit, sir?
Okay. So interesting. So we were considering at some point of time we were saying steroid is the drug of choice. There are even court verdicts on that. Then came IVIG. There are some textbooks which we studied says IVIG is the drug of choice. Then came cyclosporin. In the last Cochrane review said cyclosporin is better than the other agents or steroid. I think recent textbook, some of the textbooks also said there is some evidence for cyclosporin. I think last edition of Rook or IADVL also mentioned that. Even Indian guidelines also discussed cyclosporin, if I remember correct. And the latest review now, it says, if at all there is some evidence that is etanercept, even that is a very low level of evidence. There is no real data to support anything else. But looking at numbers, etanercept patients had some adverse effects. But again, we don't have data to say that those adverse effects were severe enough to withdraw the steroid because there's no data. That's the situation. So what will we do when we get a patient with breast STM? So this is the evidence. And how to use this evidence is the next question. So when we use this evidence, okay, most important area is evidence. Okay, if there's an evidence to say that this is better or better than the other one, definitely we should use that if possible and feasible. Okay, so here if we cannot use cyclosporin, if we cannot use IVIG, we need not regret. No, that's probably the first thing I'll be comfortable with, because there's no evidence for that also. Should we use steroid? Actually, there is no evidence to use steroid also, which we were discussing for last many years. And actually, one of the guidelines is so you don't use it for more than 3 days. Even for 3 days, is there any evidence? So far, no. Probably we can do another study or CD for 3 days and more than 3 days, then only you'll get an answer. So how to use this information for clinical use in our patients? One thing, most important thing is not to use anything which is harmful, like high dose steroid for a long time, high dose cyclosporin for a long time. If there's a patient with disseminated tuberculosis, developed TN with ATT, don't give very high dose of steroid for a long time, or any of the immunosuppressants, even the etanercept, saying that there is some evidence to help. And you can clearly say that, okay, even not using any of these agents and giving supportive care itself also can be okay. Or if at all, and there is no, again, we should think, what are you looking at? If you think there is no risk of mortality, because the mortality now over the years, probably because again, there's no evidence, probably because the improvement of the general care in our wards, the mortality has come down in STS. Now, even though we say it's a potentially lethal, many of the studies shows less than 10%. So when mortality is not an issue, we don't expect a mortality, don't give unnecessary immunosuppression. That's another thing we can, probably this study is telling us, because there's no evidence to immunosuppress, very high immunosuppression. And if at all using, probably we may have to use etanercept. That is which I would understand from this study. So anybody else can contribute, please? Dr. Ashokan is there, or Dr. Joe, I think, Joe Thomas.
Yeah.
Okay.
Can you hear me?
Yes.
Yeah. So I've had quite a few Stevens-Johnson syndrome cases back in, Lissy Hospital. So I've had a few of them. And I am a little skeptical about giving immunosuppressants because this is not pemphigus. Pemphigus as a pathology has a continuous profile of autoimmune disease. But STS is once you stop the medicine, that culprit medicine, the bullous lesion subsides. So the question is, should we still continue the immunosuppressants, or are we just giving it just to make us feel like we've done something? Because I feel like we are not focusing on the main thing. The main thing here is that there are a lot of erosions on the skin. Our focus was try to get the skin back to normal as quickly as possible so that the stay of the patient in the hospital will be reduced significantly. So why are we not focusing on more advanced methods of trying to get the skin back to normal? Whatever skin that was lost, we have to make it come back to at least one layer of epidermis as quick as possible.
Yeah, true. I agree with you, but there are two things here probably. One, there's no evidence, or the evidence for immunosuppression other than etanercept, even for etanercept, is very, very limited. Okay, there's no doubt about that. If from whatever discussion we had, that was
Okay.
continued to be over years also, as far as evidence is concerned. But does any other intervention will help? Again, is another question. Probably we may have to do another study as far as evidence base is concerned. Theoretically correct. Definitely, what we have is a better skin. Give some sort of skin. Is probably theoretically is the best way of doing it. But will it work to reduce the mortality, which is already 10% or less than that? Again, it's not very easy to answer because what we are looking at is rare disease with a less than 10% mortality. Used to be 30% at some point of time. Now it's probably some of the studies say it's only less than 10%. Our experience also is that. So how much sample size will we need to assess a significant difference in the mortality in a low mortality condition? So it's not very difficult. We may not get an answer for that. But I totally agree with Dr. Joe about the issue of immunosuppression from the evidence point of view.
And our next question, next question I have is this Cochrane criteria. How many of us actually use it? The Cochrane criteria, the age, the sex, this thing, the serum bicarbonate, serum urea. Is it actually a useful thing to decide the prognosis of the disease, Stevens-Johnson syndrome? Is it useful in real clinical practice?
It was validated in some studies, but whether it really can be translated to Indian scenario, I don't know. I doubt.
Okay. And next question I have is this thing. See, the thing is right now here in Kerala, I was actually shocked because I studied in Karnataka. We never did this before, but here in Kerala it's very common. We use a banana leaf to apply over the eroded skin. When I first joined LISI, I was very shocked when I heard that, when I actually experienced that. I was very skeptical because banana leaf, we don't know what bacteria is sitting over there. How many of us actually use the banana leaf to cover the eroded back?
No, there are two answers for that. One, I don't think there are any studies or any publication on banana leaves, but that comes to the lowest level of evidence. When there is no evidence, lowest level of evidence is the highest whatever we have. Something like experts' opinion, case reports, we all used to use. But one thing we used to do is, it's not the raw banana leaf. Usually we sort of water just warm it and wash it with some kind of maybe potassium permanganate or something like that, and wash it again to make sure that there is no crystals or no chemicals left on that. Then use it. That's what we used to advise. Whether it works or something else works, we don't know. But patients are a little comfortable to lag. So where the outcome which we are assessing is not the recovery or duration of disease or anything. What we are looking at is the patient's comfort, especially to get up from the bed. If you can, probably we can assess, can have a score, kind of scoring system to ask the patient whether are you better with the banana leaf or are you better with raw cotton bedspread. Probably it is better with banana leaf. It's not to improve the aim there is not to improve the healing. It is only to make the patient a little more comfortable. Okay.
Yeah. So I have quite a lot of questions actually. So the thing is, since we are making the patients spend so much money on immunosuppressants, cyclosporin is not cheap. Etanercept is definitely not cheap. And since we are actually working on the theory that this is not actually it's a very temporary immune response which will stop once drug has been suppressed, a culprit drug has been removed. Rather than spending this money on making the patient invest in immunosuppressants, can't we switch over to biomembranes to cover the already existing erosions, such that this
for
can
that,
help?
In a Cochrane review discussion, we need studies for that also. I don't think there are any good studies for that also.
And also, one more thing is this is a very big question which I've always been rummaging through my mind. These SJS patients, especially the TAN patients, where should we place these patients? Should they be placed in an ICU or in a burns unit? Because aren't they okay? It's little bit less than compared to a burns patient, but they are still. You can compare it to a kind of burns involvement, right? We are involved, saying quite significant amount of skin is gone in this case.
So again, I don't think there are any studies comparing exactly similar patients in burns units and in dermatology wards. But so when there is no evidence, you use common sense.
Okay.
Common
Okay,
sense
so-
is not an evidence, but it's only common sense. Expert opinion maximum.
So you feel that keeping the patient in the ward itself will be sufficient in the ward itself and let him get
I
the.
didn't say that.
Oh, okay. No. I didn't say anything. I just asked. So we should keep in the ICU initially, and then once the patient
I think
something
it
is
depends on the facility you have.
okay.
Okay, and how much patient can afford and how bad is the TN? There's no evidence. It's only common sense. If patient cannot afford anything, no point in discussing on burns unit. If patient says, okay, we can afford any amount of money, it's not an issue. I need the probably best, even though there is no evidence, probably best care, I can put him in burns unit.
Burns unit. Okay.
Okay,
sir. Thank you.
something. He's waiting for something. Yeah.
Okay. Thank you, sir. Thank you.
Ajit, shall I?
Yeah, sure.
Okay. Now, first of all, not going into any of the clinical discussion pathogenesis. No, because I think this is our focus is more on this systematic review and how do we take this evidence from this review. So I will try to limit as much as possible to that aspect only. The points that I want to tell is one, whenever we look at the evidence-based medicine, which is best exemplified by systematic reviews and Cochrane review, one point has to be made very clear to, especially to the beginners. The seniors may be knowing that, experts may be knowing it, but the beginners should really understand the thing that absence of evidence is not evidence of absence. That is a big distinction between these two. So the why I am telling that is because many a time when we do this type of exercise, even the reviews themselves admit they don't have sufficient number of good studies, and so they even collect the data. It's better than doing one study. It's always better to have a systematic review. No question about it. But even after doing the systematic review, this problem will still remain of lack of sufficient numbers or lack of sufficient studies. I am yet to find, or maybe very, very rarely, we have found the reviews which finally claim that we are satisfied with the systematic review, because most of them finally end up with, especially in dermatology, their conclusion will be, will have that rejoinder that there are quality studies are not there. We need to do more studies. So, but that is okay from when you analyze the evidence and critical appraisal part. But the patient is coming in front of us, and we have to give the treatment. So when you do that, when there is not enough in the systematic reviews or meta-analysis, you have to fall back to your clinical experience judgment, whether we like it or not, because you cannot ask a patient to wait till the systematic reviews are all over, then we start decide on the best management and start. So that is definitely not possible. So that point I am not telling it for you, Ajit, but I'm telling it for and maybe for not for the senior faculty, but especially for the beginners. So the onus is on us to have, because we are not only expected to be clinicians, we are also expected to be researchers. So in the long run, what we have to do, this type of reviews will give a indication that there is a lot more work is to be done. So that whether we can do it or not, at least we understand that. And we have to manage with the best of the best available evidence. That is a point, first point that I want to tell about this, because that is how these steroids or cyclosporin people use and still use it with conviction. We cannot even tell, we cannot tell that it is evidence-wise supported, but we cannot even tell that it's not evidence-wise not supported also in many situations. So we have to finally fall back on to something, and that's where I think the importance of treatment guidelines will come, because when you have treatment guidelines, what they do is that they will give a space for the systematic reviews and meta-analysis, Cochrane review, but they don't stop there. They stop, they continue, and if there is no real evidence, they have this recommendation based on experience and expertise will all come, and there will be a what is a comprehensive package in which you can really manage a patient somehow. Of course, we may have to deviate from that once in a while. It's one point that I want to tell. One more point I will tell about this particular thing. That is, I went through that Cochrane review summary. Unfortunately, I did not get the full text. I don't know why, because we are expected to get it.
Be accessible, it's accessible through little roundabout.
Complicated. So I did not. I did not have that patience for that. But I am just stopping with that summary only. In the summary, the etanercept versus corticosteroids is the one where they have said that they have some evidence, or they give something concrete in this review that etanercept may be better than corticosteroids in a summary. But I'm not really convinced about the risk ratio and confidence interval given in the summary, because the risk ratio is 0.51 and CI is 0.16 to 1.63. So, and 91 participants. So based on our usual sense of statistics, this does not give anything to be told significant. So, and that is one thing. And even they mentioned that it's low certainty evidence. More concerning for me is that CI 0.16 to 1.63. But they, in the summary or their final conclusion, that is one thing they have mentioned that may have some evidence in this systematic review, even though it is not also, they say that it's not conclusive. But that is the point that I don't really understand why it happened so. These are the 2 points I would like to make.
Yeah, two things. Probably again, anyone of you can correct me. When systematic review or Cochrane review says there is no good evidence for anything, probably we can step back one level and look for the forest plot and see what, just like Dr. Ashokan now mentioned, see all the studies independently and see anything looks like having some evidence. Okay, for example, one of the studies shows that it has, not in the meta-analysis, but only at the RCT level, if something shows some evidence, probably we can use it in a clinical scenario saying, okay, there is no evidence in meta-analysis, but still at a lower level, there is some evidence. Probably, since I am stuck, probably I'll try to use it when I am stuck with that. But again, only in patients who matches with that study. Not for, it may not be extrapolated for every patient in that scenario. Probably if the study is only in adults, and we may not be using that information in children. Or some kind of extrapolation of whatever is available probably is possible in a clinical scenario. So when you use the evidence, okay, evidence-based, it's not evidence-based, actually evidence-informed. You know what you are doing. Okay, so evidence is not absolute, but probably this is what we have, we use that. It's not like we are believing that what is given in textbooks or experts. We know that it is not the best, but we don't have anything else since we are using it in a particular scenario. which fits to the clinical scenario and for the patient's scenario and patient's values, etc., putting everything together in that scenario, if possible, we can. So, but we know why we are doing it, not like something like a faith-based application, because the drug of choice for TN is IVIG, the drug of choice is cyclosporin or steroid. No, we are saying that, okay, the evidence is not very high, but in this scenario, since the patient is sick, putting the risk in this scenario, I may use it. But next patient, will I use it? That depends on that scenario. So that is one thing. Again, in this study, I agree with Dr. Ashokan, but they clearly say that they are not something like promoting etanercept as this thing. They are saying it's better than probably the other ones. None of them they don't have even data to say that one is better or worse. They are not saying what is better or worse. They are saying they don't have data to say that. Most other interventions. Anybody else? I saw Suja madam. Madam, there? Or anybody else?
The Najiba madam is there, sir.
Najiba madam.
Dr. Rajiv, this is Dr. Rajiv. Can I speak?
Sorry, sir. പേര് കാണാനില്ല. iPhone കാണാനില്ല.
Thank you. Thank you. I also agree with the general trend of the discussion that the current knowledge and the Cochrane review still leaves it at the same level of knowledge that we probably had a couple of years back, except that, as you said, maybe etanercept may have some advantage over the other drugs, which needs to be approved by larger number of studies. But at the end of the day, after having gone through this Cochrane review and all, so how are we going to manage our next patient? That is the question that needs to be discussed also. As Joe said, the best way would be to identify SGSTN at the early stage, remove the culprit drug, and give the best possible skin care and nursing care. That would be the most important thing. And then our experience, we have had experience of only using steroids in the beginning, later IVIG and cyclosporin. We have not used etanercept so far in this case. And in my opinion, these, if at all, they have to be used in the early stages of the disease and for short duration, as you said earlier. So that being important, they may have some benefit if those criteria are kept in mind in the early stage. And while analyzing the Cochrane review, as you said, though we do not have evidence against any particular mode of treatment, we may have to use the knowledge that we have gained from different studies, and depending upon the cases and the scenario, whether the patient has got other comorbidities, what stage he has come, how much of the body surface is involved, we can select our drugs. And one more thing about Jones using the banana leaves. Over here, we autoclave the banana leaves. We wrap it in cotton cloth and autoclave it, and we use. The patients are comfortable with that. They are only used on the bed where the patient can lie on them, so that there is not much sticking of the eroded skin, that peeling of skin to the sheets where the patient is lying. These are the points that I wanted to bring up. Thank you.
Thank you, sir. One thing, especially for juniors, not for senior people like, senior professors. One thing we should learn to address when we make a clinical decision using whatever evidence or recommendation is, we should be very clear why we are using something. Many times what happens is, okay, treatment for Steven Johnson is cyclosporin, treatment for Steven Johnson is steroid, or treatment for a particular disease is this. But if the Steven Johnson comes to you with all the damage is already done after 10 days or 15 days of skin is peeled off, most of the skin is peeled off, then none of these immunosuppressants are not going to work. Just because etanercept came in the Cochrane review, no point in using that. Probably, like Sir said, probably on the first day, second day, it may be working. But again, we don't have any evidence for that because unfortunately, those who did the study has not reported that, whether they can reduce the total admission days of admission. There's no evidence. So, but so why exactly are we using it? Even when you use this evidence, that's why the first step in the Cochrane review is splitting up the question into different sub-questions. Like in this, what they look for is not whether these drugs are good for TN or not. That was not the question. Question was, does it significantly reduce mortality? Does it significantly reduce the hospital stay? Does it, which drug has more side effects? Are these side effects severe enough to reduce, to stop, discontinue the drugs? Like that, they're splitting up the issue into multiple small questions and trying to answer by evidence to each and every question. So when we give something, we are clear that we are giving this drug for this. Even etanercept in the study says probably reduce the mortality. That's all. There are no evidence for other points, but probably increase the side effect. That's what actually this study says. But they don't know whether the side effects are severe enough to stop the medication, because there's no data on that. So this is something very important for especially juniors when you start giving prescription. Why are we giving this particular component of that prescription? Is there any evidence to say that this component of prescription helps in this particular symptom or this better outcome? So that I think such exercises will help you to understand individual issues in a patient or in a scenario separately and tailor-make the treatment. And so one thing regarding the guidelines. So if there's a very good meta-analysis or Cochrane review, we don't need guidelines because there's no doubt about that. So that's why now many guidelines using Delphi method is coming. Because Delphi, what they do is similar exercise they are doing to split up questions into different questions and get a consensus from the so-called experts, if possible, all over the world or all over the country. So it's better than one expert's opinion. It's an assimilated opinion of various experts, which is accumulated in an objective way. So they're trying to make a subjective thing more objective. So many of these guidelines are coming up like that now. So okay, I think
can I have a small word once?
I think
Yeah,
so.
very just a repetition of what I told earlier. But please have a look closely at that particular study of etanercept, because it can be interesting, because the way it has been given, it has got a CI which should not be significant. Only one study is there. There is no pooling actually. They have mentioned there's only one study, 91 participants, low certainty evidence, RR 0.51, CA varying from 0.16 to 1.63. That's a just it is almost one is in the right in the middle.
Right in the middle.
So I'm little surprised, because the full paper is not with me. I cannot tell more about it, but if possible, I think it's better for even the presenting PG, also to have the resident to have a look closely at that particular study, and then it can be a good learning experience in that way. So I think we need to, because it's a Cochrane review, cannot be a mistake. That I don't think it can happen because it has such a big system actually. The way we understand it may be little, maybe maybe wrong. So that can be a good learning experience, I think.
Yeah,
That's
probably
what
we
I
have to
understand.
read between the lines also. Cochrane review with that their system, it's can't say they won't make mistake, but it's very unlikely because it goes through various levels of checking and cross-checking and balancing at every level. So, yeah.
Ashokan sir, I have a doubt. As we are not certain about the evidence, which recent guideline we should follow in management of STN, sir?
Okay, so tough question. We go better for it's practically it's a practical question. So when you consider that, it should be something that protect you also. Of course, go for the national guidelines.
So I think the latest guideline was in 2016. It was published in 2016 only. After that,
Yeah.
any guideline is there, sir?
So that's
I
the
don't think so.
problem
I
because
don't think
Cochrane people, produce evidence much frequent than probably Indian guidelines. So you can see what happens in MMWR CDC. Every year they come out with modifications or small paragraphs, corrections, addendums in their evidence and in their recommendations. But that doesn't happen in our scenario. Western guidelines also doesn't. Yeah, so it takes time.
Your point is very valid. I think we need to be updating whatever guidelines which are available. I totally agree with that.
So we will go with that 2016 guideline.
No, at present, you have no other option. Actually.
Okay, sir.
Even the guideline also, it's not a rule or anything.
It's only a guideline.
Also, that's why it's only a guideline. So we can definitely deviate from it, but with a reason or with why we are deviating, we should have an explanation why we are doing differently. That's all. Provided that is done, you can still manage a patient as we think it is better for a given patient.
Okay, sir. Thank you.
Sir, one thing this article forgot to mention is which of the immunosuppressants are effective with the least amount of side effects. That is one thing that I wish this article had mentioned actually. Have I missed it or have they mentioned it?
No. This Cochrane review doesn't look things like that. What they are looking is what is the evidence? There is no evidence. Cochrane review only look at the evidence. So if you ask a Cochrane review what's the evidence for parachute to save life, they will say that there's no evidence because there are no RCTs. Okay. Or what's the evidence of for paracetamol being an analgesic or antipyretic? They will say that there is no evidence because there are no studies on that. It doesn't mean that you should not use it.
Yeah, that is what absence of evidence does not mean evidence of absence. It can still be there. Okay. May I add one small point about that Cochrane? I think there was a question on that. The Cochrane, we have an advantage in using Cochrane that in counseling the patient and for those purposes. When a patient had a high Cochrane value, that means there is a poor prognosis that can be documented. So something patient does not record, that way it can help. Provided we have sufficient material to get the Cochrane done.
Sabina madam? Anybody else?
Sabina madam,
Sir,
no questions in chat box, madam.
Okay.
no questions in chat
Rajiva
box.
madam, seniors, Suma madam, Suja madam.
Suja madam was there. The Suja madam and Najima madam was there initially, but now they are not to be seen.
Okay.
Sir, I have a question.
Yeah.
Bindu. Sir, Bindu ആണ്.
പറയൂ.
As when we are in the age of litigation, so when we do undertake a treatment, can we fall back on the Cochrane review as a support for a treatment?
Yes, litigation is a different ball game. So because we know there's a very famous case as far as TN is concerned, where probably we have the highest compensation in India, 12 crores or 14 crores. So are they going to ask, no, if the question is only about giving steroid or not giving steroid, or giving etanercept or not giving etanercept, and if you can sort of argue all the methodology of Cochrane review and say that this is the evidence, and court is going to accept, good. What happens in court probably most of the time is there will be an expert. Court will not sit and analyze the Cochrane review. Court will call an expert, either court expert or our opponents will bring an expert, or we will bring an expert, depending on the scenario. So court usually will believe the expert if court thinks he is an expert. So in that case, what happened is somebody from Australia came, that particular case, and said it is not triamcinolone injection, the treatment of choice for TN. That's what that happened. And there were a lot of other issues also in that case. A lot of what do you say, that inadequate treatment, evidence for inadequate treatment, and a lot of other issues were there. So that's what happened in that case. So if the expert says that whatever we gave is a treatment which an average dermatologist will give, usually a civil court or even criminal court will not find fault with us, especially with all the recent Supreme Court verdicts. But again, consumer court is again a different situation. There it is not the legal experts. So they probably take things in a little different way, because they look only for the compensation to give kind of thing. So the answer, I don't know, but most of the time it depends on the expert says. Unfortunately, if the expert is somebody who believes only in steroid, then you will have a problem.
So it would be safer to follow guidelines, clinical guidelines.
If somebody says this guideline is old, 10 years old, and there are new evidence, so I don't think there'll be an answer. Depends on us and our advocate, and probably the expert witness.
Because in medical colleges, even now we are not graduated from the early treatments. We usually give steroids and only very rarely we give cyclosporin.
Yeah, I know. That's what I said. That's what I said. As of now, so for steroid, the evidence is very, because even the guidelines doesn't advise steroid, except probably on the first 2-3 days of the week.
Yeah.
So if a patient develops a steroid-induced problem, it's a little difficult to substantiate that. But if you gave a cyclosporin, probably the previous textbooks or the Cochrane review or the guidelines, yes, suggest that. If you give etanercept, patient didn't develop any problems, you're safe. But if patient develops an etanercept-induced problem, then still, I don't know. So that much risk anyway our profession has.
Okay. Thank you.
Any other questions or points to be clarified? So Mary,
Yes,
Dr.
yes,
Mary.
we can
there are
conclude.
no other- Okay. Okay, thank you. Thank you, Ajit sir, Ashokan sir, and Rajiv sir, and Dr. Joy for the very comprehensive and exhaustive discussion on this. This is, I think, the last Cochrane review on SDSTN was in 2002. This is 2022. The new Cochrane review. We are still at the same. Only thing is that etanercept has come into the picture. So, like Ashoka sir has actively pointed out the risk ratio and the confidence interval, the wide confidence interval, so that so in the article what they're saying is that um, the risk of mortality with corticosteroid is 163 per thousand, and the mortality with the etanercept is 83 per thousand, sir. And they have given only a very low certainty of evidence, sir. They are
Yeah,
not saying. I think it's
yeah, I agree.
yes, sir. So can we say that, sir, that is etanercept is better? That's my doubt, sir.
Not seeing that particular line. I
Okay.
don't know. Maybe that is why. Maybe it will be worthwhile to have a full article, look at it, and maybe we
No,
understand
sir,
it
in that full
differently.
article that they are saying the evidence is very low, they are saying, sir.
But
They're
even
comparing the
is it
mortality.
no, is it a new code just very low, or is it rather no evidence at all? That when we see that you see that confidence interval range
Their definition is great. According to great working group, low certainty means the effect estimate is very limited. That is what
I
they
don't
say.
think reading that I am not really very convinced about any advantage about that. But maybe I'm missing something there. I'm not very sure.
So thank you all, sir, for actually participating in this. And thank you, Dr. Farsana, especially for making a very clear and concise presentation on this topic.
Thank you, madam.
Thank you all the participants.
So thank you, madam. Thank you all. We actually took half an hour extra than planned.
Yes, sir, but that was very effective, sir, the discussion.
Okay.
Thank you, sir.
Thank you.
Thank you, madam.
Thank you.